this is part 2 of chapter 21. So let's begin. So let's move on to this part of the question, which is explain why yogurt has a pH below 7. So yogurt is actually made from milk. So the bacteria converts the lactose in the milk to lactic acid, which is an acid, of course, and therefore it has a pH below than 7. So you have to basically correlate this idea with the text that they've given you over here above, and they actually gave you a hint that they actually it's made by adding bacteria to the milk, and the bacteria change sugars in milk to lactic acid. So it's more of like a free mark question that you guys could actually think about it properly. That why does the yogurt have a pH below than 7? So of course, once the lactose is in the milk is broken down to lactic acid, so it will definitely have pH below than 7. So part B. Right, so suggest why the pH of all the yogurt samples fell slightly during the 15 days of the experiment. So if you see the table over here, the pH actually, it's still reduced over the period of time. So one possible explanation could be that the bacteria were continuing to change more lactose to lactic acid. So that is why the pH continuously fell. So when bacteria are actually constantly converting the lactose in the milk into lactic acid, so it will actually become more acidic actually. So that's why the pH actually fell, and this could be one possible reason. Another thing is somehow related to this. Now part C, the researchers concluded that adding stabilizers did not affect the acidity of the yogurt. Explain whether you think the data in table 1 support this conclusion. Right, so the changes in pH are actually very small. The greatest change is in the yogurt that did not contain stabilizers, where the pH fell by 0.5 from 4.1 to 3.6. The changes in the other 3 experiments were even smaller. So actually, for this part of the question, this hypothesis that they mentioned over here, you cannot really say that it did not affect the acidity. It actually affected, although it's a very small amount by adding stabilizers, but still the pH changed. It still changed, but slightly, but it did change. You can't say that it didn't change at all, but it did change actually. Right, so part D, using the data in table 2 and the information in the introduction to the question, explain why yogurt makers in Pakistan now add stabilizers to the yogurt, whereas this was not necessary in the past. So now you could say, in the past, yogurt was consumed soon after it was made, so it did not have time to separate. Nowadays it is transported long distances, for example from city to city, which could be a distance of like probably 6-7 hours, who knows. So because of that, it's not consumed as soon after it's manufactured. So the table shows that untreated yogurt has separated to a value of 7.9 by only 5 days after manufacture, and to a value of 21.1 after 15 days. Adding stabilizers slows down separation, so the yogurt is probably more pleasant to eat after being transported. So basically you have to say that you have to explain why adding stabilizers is actually more necessary now than adding in the past. So you have to correlate the information, the effect on the separation, and right, it's quite simple, it's not that complicated, but yeah, it's a bit of like analytical thinking actually. Mostly these questions, I mean, what I'm seeing you from my experience of solving paper 4 questions, they don't ask like very challenging type of questions like this. They don't ever ask that. This book question, some of the questions are definitely really hard, like this part, like this whole page, but like in your paper 4 question, they don't ask so hard questions like this. Never. They are difficult questions, but not to this level actually. Right, so part E. Name the stabilizer that worked best and use data from the table to support your answer. So cornstarch worked best. Why? Because you can see the statement, the larger the number, the greater the separation. If the greater the separation, meaning it's not in a good it will start tasting very bitter, very sour. It won't seem nice in appearance, so people will not buy it. So if there's like a less difference, meaning this stabilizer, which is cornstarch, is the best one. So the yogurt in which cornstarch had been added only separate to a level of 6.9 after 15 days, which is lower than the untreated yogurt and yogurts to which pectin or carrageenan were added. So if you see over here, this actually you have to find the best one. So if you make this opposite, the fewer the number, the lesser the separation. So the fewer the difference between the number in the between day 0, day 5, day 10, day 15, meaning the lesser the separation, so it will be the best stabilizer compared to this stabilizer and this. Right. Now state 3 variables that the researchers should have kept constant in the experiment. The volume of stabilizer added to a fixed volume of yogurt. So if you see over here in the whole text over here, they did not mention about keeping constant. They didn't keep anything the same. So this is one thing. The temperature at which the yogurt was kept in when it was being, you could say, when the lactose was actually being broken down to lactic acid by the bacteria, and the batch of yogurt that was used. Right. So this is actually, usually they ask this in paper 6, and you have to mention about like 2 to 3 constant variables in your experiment question when you're explaining it. Right. So now exercise 21.3, which is on golden rice. Answering these kind of questions will strengthen your understanding of genetic engineering, as you will need to use your knowledge in a new situation. Look carefully at the diagram, which contains a lot of information that will help you with your answers. Many people in the world do not have enough vitamin A in the diet. Researchers have genetically engineered rice to produce large amounts of beta-carotene, which is converted to vitamin A in the human body. Beta-carotene is orange, and so the rice also looks orange or golden. The diagram shows how golden rice was produced. So this whole thing is on actual genetic engineering. So step 1. Genes for the production of beta-carotene were extracted from daffodils and the bacterium Ervena iridovara. So the genes for the beta-carotene extracted from daffodils and the bacteria. So 2 sources. Step 2. From the daffodils, from the flowers, and from the bacteria, so you get the gene. These genes are inserted in plasmid. These are the plasmid. Plasmid is actually in the cell in the human cell as well as the animal cell. So the plasmids are inserted into the bacteria called Agrobacterium termiferans. So the plasmid, so these genes are inserted in the plasmid. This plasmid containing these genes are inserted into this bacterium, and these bacteria naturally infect plants, and so could introduce the genetically modified plasmid into the rice cell. So this cell is eventually inserted into this plant. So the bacteria is then mixed with the rice embryos in the petri dish. Some of the embryos were infected by the bacteria carrying the beta-carotene genes, and then the rice embryos now contain the beta-carotene genes. They grow into adult plants, and then they produce seeds containing beta-carotene. So this is how the genetic engineering is done. You know, it's actually quite simple over here, but in A-levels, like, it's much more in depth, the whole thing. Over here they just outline like a brief summary of how this all is done, but this is actually quite simple to understand for your level. So on the diagram, indicate one step at which restriction enzymes would be used. So this is where the restriction enzymes are going to be used. And restriction enzymes cut DNA to leave sticky ends. Explain what sticky ends are. They're actually unpaired lengths of DNA. This is like one basic statement you guys can remember. Now explain why it is useful to cut the DNA so that they're sticky ends. It's more of like forming the complementary shapes, just like enzyme substrate is basically from complementary shapes. So the same thing applies over here as well. So the base of the sticky ends of 2 pieces of DNA will pair up with each other as long as the unpaired bases are complementary to each other. this enables the joining of the DNA of the plant genes, the DNA of the plasmids. Right. So now on the diagram, indicate one step at which DNA ligase will be used. So this is that step where DNA ligase is gonna be used, and this DNA ligase actually helps. It somewhat helps in the insertion of these genes in the plasmids. And explain the role of plasmids in the process of engineering golden rice. So the plasmids are used to transfer the genes from the daffodils and nirvana into the agrobacterium. Quite simple to explain. What's the role of plasmid? And right. Don't get confused between role of plasmids and why are plasmids used. They're totally different thing. They're not different, it's somewhat similar, but don't confuse actually. Explain why agrobacterium termifications were used in this process. Agrobacterium naturally infects plant cells, so it was able to carry the plasmids, carrying the genes from the daffodils and nirvana into the rice cells. So why only specific this bacteria only? Why not any other bacteria? So naturally in the nature, this bacteria infects plant cells, so there's like a very plant-friendly cell that the scientists or the researchers prefer using this in carrying out the insertion of the beta-carotene production in the plants. Now, the last part suggests why the researchers decided to produce golden rice by genetic engineering rather than by selective breeding. Oh yeah, you might be thinking, why the whole thing just genetic engineering? Why not selective breeding actually? Right, so with selective breeding, you can only build on variation that is already there by selecting organisms with the best features for breeding. There is no natural variant of rice that has genes for making large amounts of beta-carotene, so there was no real starting point for selective breeding. Instead, genes that were already present in other species had to be used. So basically, in simple words, the amount of beta-carotene is really few present in the rice plants. So selective breeding is more of like using the best characteristics that are already present in nature. They're already present. Beta carotene, not all rice plants have that. Some of them have very few amounts, and there is no single rice species naturally that exists that has like a huge amount of beta carotene present in it. So that is why using selective breeding, it doesn't make sense in this scenario at all. In this case, you're using a gene, you're using a characteristic that's being extracted from other sources to be inserted to another third party. You get what I mean? So this is like genetic engineering. You're basically altering the features in the DNA itself. You want to add something DNA, you want to remove something DNA. That's genetic engineering in simple words. But selective breeding is more of like selecting certain characteristics of organisms that are already present in nature. Beta carotene, it's not present in nature of rice plants in very huge amounts. So that is why researchers, they use this genetic engineering process to make sure there's beta carotene present in rice plants, so that there's a good amount of vitamin A production. And right, it's basically the whole thing on that. So we're actually done with this whole chapter, and if you guys think that I need to improve on something, or if you guys think that I made any mistake, then you guys can mention in the comment section below. So if I deem it necessary, I could pin your comment. And till then, stay safe and all the best for any upcoming tests, quizzes, or exams you guys may have. So till then, I should say best of luck, and right, see you then.